This is a production of Cornell University.
Thank you very much. It's wonderful to be here. This is my title. We'll talk about this stuff first, but this propaganda about some base funding we have at Iowa State. But I have to really congratulate a lot of people. Like I think there's faculties, my professors, but a lot of the students, grad students or postdocs. Cornell is a wonderful place. I was completely transformed. I did my master's degree. I did plant breeding, PhD in quantitative genetics plant breeding, but it's not good enough, right? I talk with people. It's a 2-year, my postdoc with Ed and with the Cornell that really transformed my thinking, right? It is not a easy to say opportunity because, a lot of things going on here are the leaders, a lot of debate. The question cannot be answered here. You don't expect the other places to answer anyways, right? Because this is a wonderful place. I think I got hired, we start be able to move up to my career is because of Ed's, postdoc training, big vision, thinking that there are a lot of problems still need to be resolved, right? It's not all being solved. There's small problem, technical, there's major, large scale problems. And then one thing I have to mention too is it was, I think it was one of the seminar I gave at the postdoc launch seminar. I think there was another professor sitting in the audience, Mike Scallon. And then we were supposed to collaborate as a postdoc with Ed and with Mike's group. And I think I get a presentation, maybe I did okay, Mike, right? Mike later on, when I went to K-State, Mike, engaged me into his shoot apical meristem NSF project. And then the first grant got me tenure, then second grant get me professor. so that says how much connection you can build while you're in Cornell. It could be 2 years, 3 years, 4 years. It's research, but it's a network of scientists. It's a transformation of the thinking that really can help you later on in your career. I'll talk about some of the research. My talk typically geared towards graduate students and postdocs. So the way I talk, a little bit like a teacher instructor style. Forgive me, the professors. Okay. My position is quantitative genetics and maize breeding. I modified this position title to address significant question in plant breeding by combining cutting-edge genomic technology and a quantitative genetic theory. This type of quantitative genetic theory always give people statistical genomics, outdated variance components, but it's really relevant if we talk about the, cutting-edge genomic technology. Before I left this group, Ed insisted on me have to sequence some genes and run some gel, which I appreciate a lot, because if you are somebody who just playing the data, you don't understand the complicated gene structure, developmental biology, unless you do something. You don't have to publish papers, but do something. That really changed me. At Iowa State, we have a long history of plant breeding. This is George Sprague that started the synthetic populations. Bill Russell developed the B73, which is Steve Stock synthetic. Bill started the population. Bill derived a lot of B73 and Arnold Haller, a lot of wonderful histories in Iowa State in terms of breeding, right? Not necessarily in genetics, there's others, but this is the tradition that you see Eberhard Russell, there's a joint regression analysis paper on that. We have Kendall Lamke. Later on, I just started here. And then we believe that the power of quantitative genetics, it is not the traditional, I think Ed write a lot of breeding 4.0 and then quantitative genetic genomics, some of the postdoc writing the review papers. Really, it's still a very valid framework to combine what we learn from genomics, this biotech, phenomics, statistics into plant breeding. It's provide the engine, the framework to push everything forward. My research and training of grad students really have 4 areas: complex trait dissection. We do GWAS, try to clone some genes. They're not necessarily easy. Gene cloning project is really in depth. And then there's some gene we cloned, and then genetics, some breeding strategy. And then on the side, we do study some genomes and chromosomes. Some of the study, this is in collaboration with Mike Scallon, and then this actually gave me something to talk with Voltec, right? So we talk about recombination, methylation. We don't go into details, but it's all interesting. Thinking about this thing from the chromosomes and then the genes and then chromosomes, DNA structure to humans, we can always switch to the species we like. And then, so this is actually, sometimes we have this framework to understand we do not want a student to only stay in one breeding or one quantitative genetics, one bioinformatics area. We want to have a taste of all different areas. The book I like to share is this book. I think this book, maybe some of the professors read this book in the past. It's called The Triple Helix, written by Dick Levantine. This actually really helped us from the way we start of doing this project until we published this paper last year. I actually really carry this book with me on my flight to here. I promote people, especially you're not in developmental biology, to read this book, Triple Helix. The title is, really try to understand the genes, organism, and then this environment. Because there's a lot of historical debate about this, nature and nurture, IQ, gene control, environment, social science, and the natural science. But this is a wonderful book. I do not actually, after reading this book 25 times, I do not necessarily, 25 times for sure, I do not necessarily agree with whatever this book says. But some of the insights we gain really help us understand, okay? This is the modern summary of from the genome to the environment to the phenotype. In addition to the genotype by environment interaction, there's also something called the developmental noise that give you this final organism, or you can call that phenotype on this level, right? So most of the time, if you look at the historical textbooks of plant breeding, we profile the environment effect out before we study genetics. If we talk about entry-mean-based heritability calculation, we don't even have the environmental variance, classifiable environmental variance in the denominator because we calculate entry-mean-based heritability. But through reading this book, it connected me with better appreciation of developmental biology with the environment, what is happening. So this is a joke that we say after study genetics, we also, most of the time, I think most people in the room, we talk about genetics. It gotta be genetics, right? There's other people who think about the management, environment. We say, well, the genetics, we can study this easily because maize has 10 chromosomes, right? Wheat have 3 sets of 7, 21 chromosomes. Rice have 12, right? And then all this genetic material is organized on the chromosomes. Why don't we call this chromosomes of environment, right? They look like a centromere and a telomere. I feel that there was a joke from start by Jody Edwards, but really help us understand. Let's classify everything, right? Let's say the first chromosome of it's a radiation. You can think about radiation. Let's just think the temperature, easier. The radiation is a day length cycle first. A temperature, rainfall. These are 3 major things. And then the soil type, crop type, management. But the whole framework is a epicene, is a crop modeling framework. This whole crop modeling sometimes is difficult to understand, right? It's difficult to get into that. All my lab members actually took the workshop at crop model epicene, but it's still hard to understand. But at least we agree that it's the day length and temperature. Number 2, top 2 reasons that affect the plant development. This is different from animals. This is plant specific, right? This is another classic textbook examples. I don't know if you have taken any class that's showing this picture. This is called the gyro. This was done a long time ago. This picture is supposed to show 3 terms: reaction norm, phenotypic plasticity, and genotype by environment interaction. on each x-axis here, there's 7 different cuttings, clones. They were planted in low elevation, medium elevation, and high elevation. And then the flowering and plant, they all flowered at a low elevation. And then plant height ordered in descending order. But you see that this picture was supposed to show that there's so much G by E so that there's no clear pattern. You would not be able to predict the performance of a plant just because genetics, right? Because the same plant told us here is even got stunted here, is even not flower, right? It's here, it's here. But then you go to the high elevation, they change again. So this is showing this 3 term, reaction norm, phenotypic plasticity, genotype by environment interaction. If you have a numerical representation, they would show this lines. This is the low elevation, medium elevation, 7 segmental lines going up and down. This was supposed to be used by this Dick Leventon to show that it's difficult to track genetics or environment separate, right? But there was actually hidden pattern behind this whole thing. If you Photoshop, resorting, we will skip that, but this is just to show that context. And then this experiment was done in 1948 for younger scientists or grad students. Look, in 1948, when it's published, it says experimental study on the nature of the species, environmental responses of the climate races of Achilea. So that was done a long time ago. This elevation is actually in California. It's in Timberline, and it's, this is the high elevation, Manther, medium elevation, low Stanford is low elevation. You look at this paper title called Evolution in Changing Environments, right? The environment was changing a long time ago. And then this is a classical book to show this 3 important terms. And then this, another picture was used in the textbook, but typically this was taken from one of the early studies of Wilbert Russell from Iowa State. It just to show that the 2 maize varieties, this variety 1 has a faster response. This variety 2 has a slower response, but in general, it has less plasticity, but more stability. Farmers would prefer variety 2 over the variety 1. Variety 1 was an older variety, unless it's under the very favorable condition. Other than most of the time, farmer will prefer this one. And there's also in the plant breeding genetics, there's a Eberhard-Russell model, stability parameters, joint regression analysis. Earlier, there's other models published in Australia, but that this picture shows the stability analysis. And then when we actually study this G by E, we actually look at some of the paper written by historian. One of the historian wrote a paper called R.A. Fisher, Lancelot Hogbin, The Origins of Genotype by Environment Interaction, right? It's published in 2008. In this paper, he cited that R.A. Fisher and his nonlinear interaction of heredity and the environment. Fisher, at that early years, his main goal is to partition the variation into genotype, environment. The leftover goes to the G by E or error. He studied mainly the human height variation. There are studies where he can partition everything perfectly in genetics, environment almost negligible. Fisher also liked to do transformation of the data so that the G by interaction will be minimized, right? This Lancelot Hogbin, his interdependency in nature and nurture, he is a developmental biologist, but he's also a statistician, right? You have to be good, strong statistic to. He actually communicated with Fisher about this nature-nurture debate, genotype by environment interaction. In one of the classical debate, so this too, because we trace back to the biometric concept of G by E to Fisher, partitioning. And we trace back the developmental concept of G by E, this D, developmental, to Lancelot Hogbin, right? We'll show you that actually, when I give talks that has animal scientists sitting in the audience, I was very proud that Fisher actually is also a plant biologist, right? Because Fisher, the Fisher 1918 is regarded the the founding paper in quantitative genetics. It's still providing the guidance to human genetics these days after this post-genomic era. But this paper published in 1923 is relatively easier paper. So that when I say easy, I say, well, now we could understand, right? Fisher 1918, I talking with Jean-Luc, I still couldn't understand. Some of you people sitting in the audience, I trust, Cornell, must understand Fisher 1918. You should be very proud of you. And then you please help your other colleagues to understand Fisher 1918, right? I started 10 times, I never finished, because you can only understand the first 3 pages, then you stuck. Okay. This Fisher 1923 is Fisher 1923 contained the first ANOVA table. This is the first ANOVA table ever being published, table 3. So Fisher was doing experiment with 12 potato varieties. I don't know if any potato breeders or scientists sitting in the audience. And with 6 different manuring treatment, so the 12 and 6. And then anything deviation from summation formula, this will be the 5 times 11, that's the interaction degree freedom. And then because Fisher has the replications, you can have the random error, residual error with this degree freedom. And the sum of squares, mean squares, and deviation. So that's the first partitioning, right? Fisher did the study. But the thing is that Fisher did this study, well, was he expecting to find the G by E in manure treatment response, right? But he actually laid out his reasoning, because if you want to do a G by E with day length, with temperature, it's difficult to control. He actually did this manuring experiment. He did not find any, because look at this. In that time, we don't have F-test. F-test was invented by Snedeker, but given the credit to Fisher, F. You see the standard deviation of this mean square of 4.22. They're not much different from 3.53. So the interaction was not significant. This actually influenced a lot of animal-plant breeding. And the animal breeding typically lush. All this school of breeders, we say we're going to focus on breeding values. We're going to focus on genotypes. We're going to ignore that environment effect, ignore that G by E. That's actually why we made so much progress in plant breeding and animal breeding, which is good. But still, we didn't understand the environmental effect, right? So this is the famous picture. This experiment was done by somebody else, but this picture was used in the letters between the Fisher and Hogbin. Very easy to understand this paper. This is the Drosophila mutans, low bar. This is the ultra bar. And then this under 16 degree and then 25 degree. Hogbin was focused on the individual development, not the population, not the 16 potato right as a whole or 6 manuring treatment. He just said, look, if you say, where is the environment effect? If you look at the under the context of the ultra bar, this is my environment effect. From 16 to 25, I get this difference, right? This segment. But under this low bar, my environmental effect is this much, right? Where is my genotype effect? My genotype effect under 16 degree is this much. But my genotype effect under 25 degree is shrink to this much. So he just asked this question. But Fisher and then Lancelot, they have different focus. They could not agree on certain things, right? So the historian summarized this biometric and the developmental origin of G by E says that in that time, the biometric tradition is to partition the variation in populations. Courses of variation, this is not developmental genes, courses of variation, right? It's how much variation can be partitioned, and then statistical approach and biometric. For the developmental biologists, we focus on individual, they focus on individual development, causal mechanisms, specific genes, and then how everything has happened, right? And then interventionists use degenerative mutants. And then this developmental part of the G by E. This tradition is still being followed. But in general, we understand this whole thing is functioning. It depending whether you want to go details or you want to describe the population as a whole, right? That leads to our study. Our study is typically not large scale, but it contains enough essential element for us to couple these 2 concepts together. I was actually, as Ed said, I was at Kansas State Maize geneticist for 6.5 years. and then we were growing the sorghum, TX430 in the P. You remember T and P? This picture taken the same day. This flower, T flowered earlier than P, right? And then we went to nursery, try to get more plant. You know, I think most people have experience in winter nursery. This here, P earlier than T. We talked to plant breeders, they say, well, that's easy, right? We understand that. Yeah, it's happened everywhere, right? This T is early here, P is early here. That's nothing new, right? But I mean, why that's happened, right? We have to ask why, right? If you calculate this is just crossover interaction, this, yeah. Then I had a position change. I moved to Iowa. Iowa is further north of the Kansas. I was expecting to see the T first, P second, but in one of the Iowa, and then this is 14, P is earlier than T. So they switched again. But just totally, so this is actually a serendipity, right? I mean, sometimes if I think I move a different place, I might not see this before, right? We might not have the discovery. And then you could plant somewhere in Kansas or Iowa. You could plant winter nursery, but you could not plant in winter in Kansas or Iowa, right? But what we talked to the crop modelers, physiologists, they ask us to say, what if you plant the same population? These are the two parents. We have a real population in Puerto Rico. We thought they're going to happen the P first, right? But what happened in Puerto Rico, T first. So they just keep changing the two parents, keep changing the orders of flowering time. A lot of people just, if you're breeding or applied research focus, you know that was happening. You don't ask why. We happen to do some genetics, so we ask why that's happened, right? We also know there's a lot of people doing the gene regulatory pathways, networks of flowering time. There's multiple pathways, autonomous pathway, gibberellium pathway, this photoperiod pathway, many of the pathways. There's FT, you have to synthesize floragen to obtain this vegetative to flowering transition. For us to grow the tropical material, we typically cover them with a trash can or the black forms to induce a flower, right? That's the early stage. We know that some of the photoperiod synthesis happened in early stage of the plant growth development. And then, but we didn't stop there. We say, let's actually show this whole picture here. They seems like, let's try to discover patterns underlying the apparently complex G by E. That's the sites. We have the experiment. This is a reaction norm plot. I think Joe Gage is an expert in reaction norm, and he published a good paper on this. This is a Puerto Rico 11, 12. This T is a blue and the red. That's the highlighted dots. The other is 250 reals. This is a small mapping population. They're not complicated design anything. It's just mapping population. You track the flowering time change. Here T is early until here P is early. P is late. This T is early and then change again, right? The traditional analysis, you can sort this by instead. This was actually arranged by the photoperiod day lengths from Puerto Rico to Iowa. But if you have the data, you order by your data categorically, you say that, okay, in these three sites, that blue, which is T, is later than P, and then things changed, right? The photoperiod temperature changed. And then, if then you numerically taking the average we call environmental mean, it's the average of the population flowering time transformed into the x-axis. This now became a joint regression analysis. So the x, y axis using the same data, it's just the x axis using the environmental mean, and then the y axis depict all individual lines. Now everything looks nice, right? It's just saying that if the whole population flowered early, and they're here. If the whole population flowered late, they're here, right? It's going line. Actually, this figure is actually nothing new because in genetics, in plant breeding, we all understand it. Joint regression analysis. There's nothing new here, right? Now, what is new is that in this general context of predictive phenomics, right? We want to understand, okay, I talk about multiple chromosomes of the environment. If we have a genotype somehow squeezed into a one-dimensional space, we can squeeze this environmental as the fertility or richness or something. How? Let's just squeeze into environment here. Now our performance is actually on the z-axis. So you actually have a landscape, right? You have the x, y, and z going up and going down, right? So we say, well, if we actually can sample this whole landscape by sample certain genotype, its performance under certain environment, we actually give a data point to actually to map out this up and down. And then we can predict what's in the future, right? So in plant breeding, we also have a term called the target population of genotype, target population of environment. This two terms just say we're mapping within something is tractable, right? You cannot throw mutants here, then it's gonna be, this whole thing will be distorted, right? So the general idea about when we had the data, we realized after many years of doing, most of the time in scientific studies, what we do is a pattern discovery. We just discover patterns. If you want your paper published, you look at figure 1, figure 2, figure 3. It just cumulatively to support your final conclusion because you see patterns. It's not random, right? A statistical test is against randomness, right? So your pattern finding is a whole process. So basically, in this whole area, we try to, plant breeding, we have the tested environment, tested the genotypes, right? Most of the time, we do not want to study G by E because we're not like companies. We don't have enough funding to have multiple sites everywhere. Within our sites, we want to be homogeneous and then we can just get some variety. But if you are in charge for breeding for large geographical regions, you do have to deal with G by E, right? So, and then this is the untested genotype, right? The key thing is for our research is to connect the tested environment to the untested environment. This untested environment can exist in the future. Let's say 2019, we haven't planted it yet, right? We have problems to get this into the field, right? Or 2021. And then this connect the tested genotype and untested genotype. This is relatively easy. We have experts sitting you can actually, if you look at one of the paper, S group published, you can connect the tested genotype to the Asian to maize, right? Can predict its performance, right? This side is relatively straightforward of genomic selection, but it's tested environment, the untested environment, you can connect with quart models, right? And I think one of the earlier papers from Heslot with Mark Soros group is a very influential paper published to say how you connect the environment together, right? What we really want is a future genotype, right? Future inbreds, cultivars, or the future hybrids under future environment. But we have to establish all these connections. So our research actually is really, this diagram was first time shown by, I think, Fred Van Ewijk. We just learned how to do this thing, right? So our goal is to connect the environment tested and untested. So we can do forecasting, we can do predicting in the future. We look at the day length, Iowa, Kansas, Puerto Rico, and we look at temperature. It turns out that if you plant in the summer of Puerto Rico, it's a long day environment. Puerto Rico is a long day environment. I thought it was a short day, but you look at the day hours, it's 14 hours, 13, 14, right? Typically, we use 12.5 or 13 hours and long day, short day separation. This is true for sorghum, also true for rice, I think, right? And then temperature, we couldn't see any patterns. We cluster the temperature, there's something going on here. If we use performance of reals, do the two-way clusters, we feel the Kansas was together, Puerto Rico, Iowa, and then Puerto Rico 12. So Iowa 14, summer planting is clustered together with Puerto Rico 11, Puerto Rico 12. But Iowa 13 was clustered with Puerto Rico 14S. There's all different patterns we can figure out. You can almost ignore this slide because we're gonna show you what happened. What happened is that we studied the temperature, like day length alone. Day length is, this is a 3D correlation plot. So day length here, day length also here, and with this average environmental mean of the population. So now we're talking about the whole population, try to correlate that performance with the day length and then temperature. The correlation is not very high. The temperature profile is not high. But if you do something called the Photothermal time, GDD times day length. Then you have this beautiful shade pattern of correlation. It's these dots going on. So basically, low temperature is mimicking a short day environment. Temperature and then the day length as a compensation between them, right? This actually is also not new because that in the crop modeling, we know this PTT photothermal time. There's thermal time, green degree days, or day lengths, but making them together. But the key thing is next slide, we search a window to find this critical window where PTT can predict the population mean. So here is actually summarize everything we try to do. So we know that in the early growth stage, so that's why we all like to talk physiologists these days. They tell us what's going on. There's a temperature thing, day length thing. Then the determination of time, the transition from vegetative to to the reproductive stage was determined here, right? And then we basically, I'll show you this search process. But here is a 2D scan. The beginning of the search window, end of the search window. Here, we basically try to calculate a large number of correlations, okay? The correlation, here we call population mean. You can think about environmental mean. Is a population flowered early or late. See, if I tell you that Puerto Rico 11, 12, they're flowered early, but Kansas flower late. So that's the dots. And the photothermal time, basically we try to find, if I start at different stage, where do I find the most correlated window that can give me the predicted flowering time in the end, okay? Don't worry, we'll show an animation to see how this search going on. Eventually, if now, remember earlier, I have a figure, I said this is nothing new because it is really nothing new. This is, okay, it's here. This is a typical, if you have the population grew out, you already done the experiment, you have an environmental mean, you have the mean, right? But what if I didn't do the experiment? So the key is to replace this environmental mean with an environmental parameter, right? So that's all the struggle we went through, try to replace this parameter. So after you replace this x-axis, now it's called the photothermal time. It's photothermal time during a spatial, specific growth stage. If you have that photothermal time, I can predict the flowering time. So that's, we change the x-axis to make it the predictive in the future, right? Let's see how, we call this called the critical environmental regressor through informed search. In the earlier papers and also the crop modeling papers, they just have so many parameters. They all get together, they're functioning. I mean, I just don't understand what is being calculated, right? But they give a lot of insights to us. What we try to take out the essence of the model, saying, among all these parameters, what are the most sensitive parameters? In this case, it's photothermal time, right? So we basically have we have 7 numbers. This 7 numbers is the x-axis, this value, projected x-axis. 7 environment, we have the means. We change the search window of the green of the photothermal time to see which one give you high correlations. So we can define a 3-day window. We can define a 7-day window, right? Every time we have a window, this is like, if you know how to program, it's very easy, right? You start and end, right? And then so when I start animation, these dots, this horizontal position is not gonna change. This position gonna not change. It's changing this x-axis window, right? That's how it started. You basically try to build lots of correlation to find where is the best correlation can replace the environmental mean, so that I don't, in the future, before I do the experiment, I know I can predict what's gonna happen, right? Here, we got, we find out it's 18 to 43 days that get calculated the photothermal time can be 0.996. It's all essentially 1, right? People ask me, how about the rainfall, how about others, right? And people say correlation, causation, right? If you get a correlation 0.996, you don't care about the correlation causation, right? That's good enough, right? Because what else you could do, right? But it makes sense to ask why that's the case, right? Because you have backup of the physiological research or developmental biology research, right? So once we have that done, we can actually do a joint genomic regression analysis through the reaction norm parameter estimation. Basically, because now you have this x-axis. Now, if you derive this x-axis from the experiment, it became performance-free. Now it's just separate, right? Unless, as long as you stay in the typical environment, you can do forecasting. Each genotype, its performance along the x-axis became captured by the intercept, if you do an intercept here, and a slope. So everything just intercept the slope, right? And then if you predict the tested genotype under testing environment, predict the untested environment, that's easy because you can read off, right? You can predict under the tested environment, you can have some genotype will have this performance, intercept slope. The other genotypes, you use gblob to predict the intercept, predict the slope, and then you just read off your result. You can hide your data to do tested and genotype under tested environment to the untested genotype, untested environment. The key here, people ask, why this correlation 0.95, 0.87, 88, 86 is so high? Because this reaction norm does not only contain G by E, but it also contains the genotype effect, but also contain the location effect. Look, everything, this location always flower late. This location generally flower early, right? So this high correlation, we predicted it contains both location prediction, the environmental prediction, plus the specific prediction for each genotype. That means if you read this picture, it say the observed G by E, right, is a natural process of the plasticity containing each genotype plus the environmental input. It show up, right? Because this is, remember before we sort everything out, it was up and down, it's crazy, right? After you sorted it, you can do predictions. It takes time to understand this, but let's see another companion thing. You can also do a genomic regression analysis with a genome-wide marker effect continuum. This is to say that in genomic prediction, genomic selection, we have two terms. One called gblob, that individual's performance was predicted through gblob, right? Through the mixed model additive relationship matrix. But you can also assign this breeding value to each marker. So each marker can give an RR blob, right? To have the marker breeding values. Here, I'll show you that this is through the marker effect continuum. So here, earlier slides, each line is a genotype, is an individual, is an inbred. Here, each line is a marker. You see the marker has its effect changing down or up depending on the input of the environment. Here, we changed the numerical, but we added here. That's where we derive this thing, right? Remember, these two values are almost one, or essentially highly correlated, right? So you can, through prediction from the marker effect, you give me an individual, I have this, I just summarize the marker effect, I can do the predictions. This equivalency between the G blob, RR blob was realized by the reaction norm parameters. I will have more slides to show that, right? Again, in most of our genomic selection paper, I think we do a lot of this, Jean-Luc do a lot of this. People always sometimes ask empirical validation, but the data is empirical. But people always ask, what about you do additional experiment, right? So that's what he did. It took a time. So remember that earlier analysis, we have the Puerto Rico, Iowa 14, and then Iowa 13, Puerto Rico 14, summer planting, Kansas. Then we planted Iowa 15, 16. Our model stopped before Iowa 15 and 16. Now we're going to predict what is happening in Iowa 15, Iowa 16, and then to see what happened, right? So the Iowa really nice because see Iowa 14 is here, Iowa 16 is here. It spans this crossover region, right? So that means the temperature and day length has an interaction itself, right? Environmental factors has interactions, and then interact with the genetics and give you this G by E interactions, right? So we did this empirical validation. The correlation coefficient is 0.8, and then it's 0.88. Here, we're a little bit off our target. That's because the window we selected is somewhat fixed. If we could, in the future, select a dynamic window, we believe that we'll have a better target. So that's the empirical experiment we ran in 2 years to prove whatever we can do, right? So this picture also facilitate you to think about in the breeding companies, right? If you know the historical average of each site, you can optimize your sites to generate environmental that optimized cover the, cover the x-axis. You don't have to testing in these locations forever. Some location might give you redundant data, right? You can optimize the testing sites. So we didn't specify this in the paper, but it's there. And then we say a lot of research we do, we say to explain or to predict. I have to go faster. Most of the time in the nature explain, we can do to explain. That means we do genetic study or developmental studies. We generate the mutants. We do gene cloning, magnetic studies. Also, we do GWAS, select significant peaks. But on the other side, we do the black box approach. We do this prediction. You can think about machine learning too, right? But sometimes there's a human nature. Even sometimes we can predict. We still ask why we can predict, right? That's why if you publish a paper saying explanatory machine learning, and people like it because they can connect with biology, right? But these two actually, it should be joined together, right? So this is the same slide about the regulation networks. So the same set of data, we can do basic QTL mapping. If you do QTL mapping, you'll see this 5 genes give you the peaks at different locations. But then you can do multi-environment analysis. You can get this one, single environment analysis. And then the epistasis is also significant. What I'm saying is here, although prediction is based on genome-wide markers, there's major genes that affect, generating this effect, allow us to be predicting. We'll see this slide again, right? These are because two parents have genome sequence, we can actually relate this QTL peaks to the specific gene that other groups have cloned. So these are the different, alleles. These are the geopics to show that when we say claim is the gene underlying these peaks, we have some confidence to say that, right? This thing is called, now you can do genetic mapping of reaction norm parameters. Like this was done in other groups too. But this reaction norm now is the reaction norm generated from an environmental index. It's not an environmental mean anymore because we believe that when you use this reaction norm generated from the environment index, any gene you showed up should be the gene that directly responsive to this environmental parameters. You can find this same thing, same peaks. The interaction also significant for the intercept, for the slopes, right? So the performance across the geographic diverse environment is a phenotype too. So you just did a transformation there. And then we read the earlier papers by Thomas Younger. His group did a investigative summary to say that sometime you can have gene effect of antagonistic pleiotropy, or conditional neutral, or differential sensitivity, or no G by E. That means your QTL can be up here, down here, detected, non-detected, both detected by different effect or none, right? So if we actually follow the same logic, we can actually say this other 7 locations, you can see this QTL is MA1 in certain locations as a negative effect versus the other allele, reference allele, sometimes has a positive effect, right? This is because we think that differential sensitivity, conditional neutral, or antagonistic pleiotropy, this all is just depending which segment or which gene you're looking at, right? It's because plants are mathematicians. They have to learn all this time integration or time averaging or the rate detection, right? They know how to do differential equations, right? These are the categorical 7 location, but if you plot this photothermal time, now we're showing this genetic effect. It's not a constant across the environment. Genetic effect is changing the size. Remember the big A, big A, big A, big A, small a, right? It changes the size and directions along this environmental parameter called photothermal time. And then this is a genome-wide marker effect also changing. I mean, this is actually true, right? Because that plants, they try to have different environmental input, they try to flower, right? But make a decision to flower. So they change their effect, right? So here we say unifying developmental and biometric aspect of G by E is enabled by genomic analysis because we think that developmental G by E or developmental everything was happening here. But if you just analyze the final results, that's the biometric G by partition variance components, calculate heritability, right? But then what happened here is individual, right? So that's got united. We made a lot of statements saying that integrated analysis of genes and environment can reveal the magnetic interplay under the observed phenotype dynamics. All this are, so this is mostly for the breeding company, large companies, right? You could actually sometime optimize your testing sites. You can also think about how to use the winter nursery observations because it's genetics, right? It's environment. If you figure out the G by E, you can leverage some information from the winter nursery data. I'll skip this. So we say this prediction is site specific. It's on target. It's whole genome. It's in season. This IS is in season. So you can actually, if you can really think about how you design and thinking about data mining, you have the data, you mine the data, you see the pattern, and then how you use this pattern to direct your future experiment, how you decide which genotype to pick. You want to pick the most representative genotypes for the whole landscape. You want to pick the environment based on historical 15-year average to testing your, your genotype, right? So we get this paper published. So we really happy about this paper. This paper contains tons of stuff we did. It's not that complicated. It's a real mapping population, and then but we were able to get this published. Does this work for other traits, other crops? So far, I work in for flowering time, plant air height, grain yield. We work on all this stuff, all these crops. We have data to work on. And this is a rice study we did recently. It turns out that it's not photothermal time. It is a temperature profile. Actually give you this negative 0.99 predictions. So this is ongoing research. And then does this work for other traits? We really got asked many times. We say, well, guess what? It's physics, right? Right? When we have these first two chromosomes is the radiation. You can think about day length and the temperature. Well, Earth's been rotating, Earth's been revolutioning around the sun and rotating by itself with a tilted axis, gives us the seasonal changes. Why, whatever people like crop modeling, why it would not work? It would work, right? You have all the plants show up on the planet. They have to adapt to the seasonal changes, temperature changes, right? They just, this is just say, well, finally we think that some of the biological observations have to agree with the physics, law of physics, right? Because that's what happened. So we also did something about optimized design of genomic selection of the hybrids, because there earlier we showed the testing environment can be optimized, but then the testing individuals can be optimized. We'll skip that. Eventually, we say instead of the to explain to predict, we believe that you can do to explain and to predict, right? So under our narrow context, we say you can establish a unifying framework for genomic prediction under untested genotype, untested environment. But if you think more, right? Think about the 3D structure. We could say there's a unified synthesis framework, at least synthesis framework, that can combine genomic selection, GWAS, and G-BioE all together. We don't want to say GS, GWAS under G-BioE. We want to say GS, GWAS, and G-BioE, because remember that 3D picture of environment and then genotype and performance. Most of the time when we do GS or GWAS, either we slice through this 3D to do our GWAS or GS, or we squeeze or summarize this 3D to a 2D, right? These three things actually can be all think about together, right? I think we work on, I learned genomics, I learned quantitative genetics, I learned a developmental biologist. I really like to talk to everybody because we can actually help us to explain, to predict, right? We got funded by different agencies, and then Xianran Li and Tingting Guoxing Li, major contributors of this research. And then we have Mike Scallon was an NSF-funded SAM project. We have other collaborations. I think that's all I have to say. That's all I have to say. Thank you very much. You have to start.
So I was, wondering, what was your power and resolution to detect the QTL when you just went by different environments rather than using photothermal time? If you wanted to do that to discover what the genes were, did it help to understand photothermal time or not? In terms of resolution and power.
That's a great question, because after the study published, we keep thinking. I think you guys are going to be thinking too, right? They say the minimum environment to study G by E is 3. So you can have an intercept and a slope, right? We say, what about individual 2 environment? Can you study G by E? If you fit your 2 environment under the context of reaction norm, the answer is yes, because it's the average and the difference. It's not as good as you have 3 dots to have a regression line, but it's a good approximation. Then go back to single environment. Can you study G by E with single environment? Most people know, because you have to have 2 environments, right? But actually, any major QTL peaks you detect in a single environment is somehow pointing to these plasticity genes. If the single environment is sitting on the x-axis of the environment index change. Your question is great, because in single environment, besides the major environment index changing your flowering time, there's some local specific environment. Let's say there's a dry spell, or let's say there's some extra nitrogen changed your environment. Then you detection power will be low, because every phenotype is shifted to the whole pathway, the network, this perceptron will shift to that signal. But if you feed that signal into the general framework of the range of the environment, yeah, it should be detected. I don't think I answered the question, but it's for us to do this research, we did do this individual and together. That help us to see why some signal persistently showing up in different environments, because that's capturing the main drivers of the environment. But other signals will disappear because other things twisted it along. Yeah. Thank you for that question.
Questions?
Oh, there.
So obviously, like for photothermal time, your day length and heat, they're huge, and they control a lot of stuff. And also, they're useful for prediction, I guess, because if you know where you're going to grow something, you can make a pretty good guess.
Yes.
But I'm just kind of curious how your other environmental chromosomes, if you will, things like precipitation, affect your predictions, or do they improve it if you know them, or are they just totally pretty much worthless in this framework?
I think they're very useful. But think about we have to get the data to give us, let's say, with a relatively fixed photothermal time of day length and the temperature, and then different rainfall patterns. So the thing is that the pattern discovery, predictive power, only if you have detectable patterns in the data could you then be able to detect them. So far, for the flowering time and also for plant height, because the experiment we ran, typically we irrigate them, or typically, unless they became an outlier of the whole other experiment, we take them out, right? So otherwise, we haven't been able to use the rainfall pattern to improve this model. But it is possible, especially for large companies. You ran a lot of irrigated trial, or if you have large data, you could improve. Or it says that because the environment is continuous, but the developmental physiology at a certain time, it's become critical. If you can capture what is happening there, and then you can have some predictive power. That's a great question. Thank you.
I guess I'm happy to acknowledge that it works. I don't understand why it works. I think you said plants are mathematicians. For flowering time, pretty clearly they have to be good mathematicians, because if you don't flower at the right time, it's bad.
Yeah.
For yields, it seems like mathematics breaks down. There's many different things, like, what if fertilizer comes now, or what if rain comes later? I mean, it's just, and so, then, I mean, the fundamental process is you take your environmental parameters and you figure out some kind of combination of them that gives you your correlation of 0.99 or better, right?
Yeah.
And then you use that for slope and intercept. But given the number of things that affect yield, there's a multiple testing problem in terms of, I mean, because you have water, but you don't have, I mean, you showed, sorry, I'm going on, if you shut me up. You showed, like, you have one interval, either 1 to 4, 1 to 7, 1 to 10, 2 to 7, et cetera. But you have one interval, but, why just one interval? Maybe there's 3 different critical intervals that determine flowering time. And right, so then I'm sure I could take just your 2 numbers of temperature and photoperiod and get a higher R squared than 0.996, right? I think you can get to 1. Okay, so then you start thinking about, all of the other aspects of fertility and moisture and solar radiation, and and they're spread out across week number 1, not week number 1, day number 1, day number 2. So there's a multiple testing burden where no matter what, even if I had 50 environments, I could get you an R squared close to 1.
But remember that crop model will handle everything you said, right? But then here we're just saying that among this large geographic region, what are the most essential components? Allow you to have the general predictions. If you do want to go to that finer scales, you do the crop model. Instead of one axis, detect that one period, you have multiple periods, right? You have all information, right? That's whatever crop model, they take everything. But the problem with the crop model is that we, as geneticists or plant breeders, we couldn't understand what is being the critical, essential stage, because the weather pattern is continuous, right? The day length won't change like this, right? The temperature might change, but the average temperature, can be smoothed. We want to just show, actually, the reason why we could, the statement you made about what if we find multiple windows, that's great, because that's what our study showed, right? So as long as we can find the windows, 1 index, 2 index, combination of index, machine learning can find some things, get the better prediction, that's great. Because that's the purpose of this paper, to say we can make predictions of the pattern existing in the data, because they're data, and then we can do the major ones first. Well, there's a paper published in Nature Genetics just last week. They did a crop model with Frank van Ewijk with the Iran yield. They have better predictions, but still you cannot tell what was being predicted. That's a hard question.
I think we'll end there.
Okay.
And we'll thank Jianming again.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.